Hello, I'm Minerva Oliver, one of the scientists at Sartorius. I will be walking you through one of our posters presented last year at SLAS, and this poster is entitled Real-Time Live Cell Analysis of 3D Tumor Spheroid Invasion. Now, before I get into some of the assay setup and the data generated using this approach, I really wanted to give a brief overview or background on cell invasion. Cell invasion, as we know, is a hallmark of malignant cancers, and it plays a key role in tumour dissemination and metastases, and has been attributed to the high incidence of cancer-related deaths. The ability of tumour cells to form a metastatic tumour primarily involves cell morphological reorganisation and degrading of the surrounding extracellular matrix, as you can see from the image here on the right. So briefly, local invasion is initiated as the signalling pathways that control cytoskeletal dynamics in tumour cells are activated. And once these cells are activated or transformed, they then begin to detach from each other due to reduced adhesiveness. And you can see here in the image that they then degrade and disrupt this basement membrane by secreting proteolytic enzymes. They then invade through the disrupted membrane, through the stroma, and eventually metastasize by way of blood vessels or lymphatics into secondary tissues. So targeting such events or some of these underlying signaling pathways using low toxicity drugs, for example, holds great potential to disrupt cancer cell progression through this cascade. Conventional in vitro invasion models, such as the filter-based transwell invasion assay, are widely used to assess tumour cell invasion. However, they do lack some comprehensive cell-to-ECM interactions, and this may really limit their physiological relevance. Here, we provide a simple 3D tumor spheroid invasion model to recapitulate key features of the metastatic phenotype, and we demonstrate the use of the IncuCyte live cell imaging system to kinetically acquire and quantify 3D single spheroid invasion. The IncuCyte system is a real-time quantitative live cell imaging and analysis platform, and it enables visualization and quantification of cell behavior over time. And it really works by automatically gathering and analyzing images around the clock within an incubator. This live cell imaging system, together with simple workflows like the one described here and non-perturbing reagents, provides ease in assay setup, image acquisition, and data analysis. So this poster really describes the use of this IncuCyte system and the validated 3D single spheroid invasion assay to study the invasive potential and capacity of malignant tumour cells. So shown here is the 3D single spheroid invasion assay workflow validated for use on the IncuCyte system. So briefly, invasive tumour cells of interest are harvested and seeded into ultra-low attachment round bottom 96 well plates. They're then centrifuged for 10 minutes, and the plate is then placed inside an incubator or inside an IncuCyte within your incubator to monitor spheroid formation. So once these spheroids are formed, the plate can then be removed from the IncuCyte system, and then spheroids are treated with antimetastatic compounds of interest and subsequently embedded in matrigel to induce the spheroid invasion. The plate can then be placed back into the IncuCyte to continue monitoring this biology. So to exemplify the assay platform, U87mg glioblastoma spheroids formed for 3 days were embedded in matrigel in the presence and absence of actin polymerization inhibitor cytokinase D, and depth of focus bright field images were then acquired every 6 hours in the IncuCyte system. So IncuCyte's bright field images shown here really show the effect of cytokinase D on spheroid invasion, and they also show the extensive invasive phenotype that was observed in vehicle-treated spheroids in comparison to cytokinase D-treated spheroids. So single spheroid invasion properties were measured using IncuCyte's automated spheroid invasion software, which tracks and quantifies changes in spheroid size. Now note this software's ability to accurately segment the whole spheroid area, shown as a yellow outline mask, or the invading cell area, shown here as a blue outline mask. And the time courses plotted here on the right show the individual well bright field area over time, and again they also illustrate that in the presence of cytokinase D, U87mg spheroids markedly increased in overall size and exhibited extensive invasive capacity. So to assess the metastatic potential of tumour cells in 3D, we also then seeded some U87, A172 and HT1080 spheroids in ULA plates and allowed them to form spheroids prior to being embedded in matrigel for 7 days. And these time-lapse videos revealed cell-type specific invasive capabilities and demonstrated the software's ability once again to accurately and consistently segment the whole spheroid area or the invading cell area across a range of cell types. Now, to account for variations in spheroid size following formation across the 3 cell types, the whole spheroid area was normalized to spheroid size at t=0 hours and automatically plotted over time. And as you can see from the time course here, at 168 hours, the U87 mg spheroids increased 2 to 4 times the size of HT1080 and A172 spheroids respectively. The invading cell area time course shown on the far right shows that though different in overall size, the A172 and HT1080 spheroids exhibited comparable invasive potential over time. Now, to understand the relationship between ECM properties, invasive capacity and invasive phenotype, HT1080 spheroids formed for 3 days were embedded in increasing concentrations of matrigel for 10 days. And the acquired images shown here and the quantification of the whole spheroid area demonstrated that spheroid invasive capacity is matrigel concentration dependent. And the bright field images also illustrate the effect of matrigel concentration on invadopodia. And upon interrogation of some of these images, as you can see in the zoomed in images below, there's marked density and elongation of invadopodia at the highest concentration of matrigel, the 4.5 mg per mil. Interestingly, area under the curve analysis of time course data revealed a bell-shaped relationship between matrigel concentration and invasive capacity of U87 mg and A172 spheroids. And though further experimentation is required to confirm these observations and to really determine an optimal matrigel concentration for rapid cell motility, others have reported that at an optimal matrix concentration, cells at the periphery of the spheroid readily align with the matrix fibers and rapidly invade the surrounding ECM, whereas at higher concentrations, cell motility is reduced by the rigidity of the matrix. And again, this type of data really shows the value of the IncuCyte in enabling you to get additional insight, not only by looking at your images, but also looking at the quantification. To exemplify the amenability of this 3D tumor spheroid invasion assay to antimetastatic compound testing, we performed a pharmacological study in U87 mg cells. So U87 mg spheroids were formed as previously described, and they were treated with invasion modulating compounds and imaged every 6 hours for 7 days. And you can see here they were treated with cytokinesin D, PP242 and blebistatin. So the IncuCyte system is able to generate these real-time automated plate views, which enabled rapid visualization and assessment of compound effects on spheroid invasion. And you can see, looking at these plate views of the whole spheroid area and invading cell area, that cytokinesin D and PP242 inhibited invasion in a concentration-dependent manner, while little effect was observed by blebistatin. A panel of known antimetastatic compounds were then tested for their effect on U87 spheroid invasion. Spheroid invasion properties were kinetically quantified using IncuCyte's bright field size metrics as I've described previously, and the representative bright field images taken 4 days post-treatment showed a wide range of inhibitory effects on spheroid invasion. And with the exception of blebistatin, which appeared pro-invasive, all compounds inhibited spheroid invasion as reported by a reduction in both the whole spheroid area shown in orange on this bar graph and invading cell area shown in grey in this bar graph. Cytokinesin D and PP242 appeared to comparably reduce the overall spheroid size as seen in this bar graph by 90 or 80% respectively. However, upon inspection of the invading cell area bar graph, we can see that cytokinesin D effectively inhibited invadopodia in comparison to PP242. So to understand the inhibitory effect of PP242, we normalize the invading cell area to the whole spheroid area at 96 hours. and this further demonstrated the marked inhibitory effects of cytokinesin D in comparison to PP2 on the invading cell region. Note that 80% of the total area of PP242 treated spheroids was attributed to the invading cell area in comparison to only 30% in cytokinesin treated spheroids. So, a subtraction of the invading cell area from the whole spheroid area region would provide a measure of the spheroid size of the spheroid body, and hence a measure of spheroid proliferation. So we performed this subtraction and it revealed the antiproliferative rather than the antimetastatic properties of PP242. Now, to validate this analysis approach, we performed a separate study in the absence of Matrigel, and again similarly PP242 appeared to inhibit spheroid proliferation as shown in this bar graph as a reduction in the largest bright field area. Now, this really shows the power of the Incucyte not only to understand the effects of these compounds visually by looking at the bright field images, but also the use of integrated metrics and analysis approaches that enable you to distinguish compound effects on spheroid invasion and spheroid proliferation within an invasion application. The pharmacology of antimetastatic compounds was assessed across multiple cell types, as you can see here, and concentration response curves representing the area under curve analysis of the invading cell area time course data from 0 to 7 days was generated. And we can see from the CRCs that cytokinesin D was a potent inhibitor of invasion, causing comparable inhibition across all cell types with equipotent IC50s, as you can see from the table below. Concentration-dependent inhibition of invasion was also observed with PP242 across all cell types. However, you can see from the U87 CRC graph that inhibition was incomplete and partial at the highest concentration, where 60% inhibition was observed. The GM6001 compound only appeared to inhibit HC1080 spheroid invasion, as you can see from the CRC. So again, this data really just shows and highlights the value of this system and how it can be used in pharmacological testing of potential antimetastatic compounds. This poster was really put together to help describe and show a simple 3D tumor spheroid invasion assay workflow that recapitulates key features of the metastatic phenotype. And in conjunction with real-time live cell analysis, I've been able to show that with the Incucyte, you can not only monitor and quantify cell type-specific temporal invasive potential, this platform can also be used to probe effects of spheroid invasion and spheroid proliferation using integrated metrics, and it provides a validated method of identifying and distinguishing new and existing compound effects on tumor invasion. And additionally, this model can be further developed with the incorporation of stromal cells to address diverse aspects of cell invasion. I hope I've been able to demonstrate to you that these validation methods and data demonstrate the ability to kinetically visualize and quantify spheroid invasive properties across a range of cell types and illustrates the utility of this model for real-time compound profiling. Thank you.